I want you to start by trying a little thought experiment with me today.
Oh, I love a good thought experiment.
Oh, right. So picture your siblings, if you have them. Or, just think about the concept of biological siblings in general.
Sure.
You all came from the exact same two parents. You were built from the exact same biological source material.
Right, 50% mom, 50% dad.
Exactly. So why aren't you an exact physical and mental clone of your brother or sister? Like, why are the results of that exact same parental equation so wildly, completely different every single time?
Yeah, it is the ultimate biological lottery.
Right.
And the physical mechanism behind that lottery, like the actual cellular machinery that creates those differences, is what we are pulling apart today.
Absolutely. Welcome to today's deep dive. Our mission for this session is to completely unpack the incredibly complex, frankly beautiful, and sometimes very error-prone biological machinery known as meiosis.
Yes.
For those of you diving into the anatomy and physiology of reproduction, that's the specialized cell division that creates eggs and sperm.
Which is just a fascinating topic.
It really is. And to do this, we are pulling from a really cool stack of sources today. We've got anatomical biology texts, some foundational historical cytology notes, and the latest modern genetic research.
Yeah, and we really need to establish the stakes right at the top here because, this isn't just microscopic trivia for a biology exam.
Definitely not.
Meiosis is the fundamental reason sexual reproduction even exists in the first place.
Wow, yeah.
It's the engine of human evolution. It is the literal driving force behind what we colloquially call the biological clock. And it dictates prenatal care, fertility, and really the very blueprint of who you are.
Okay, let's unpack this, because before we can understand how meiosis works, we have to understand why it mathematically has to exist.
Right, the math problem of life.
Exactly. It's there to prevent the human genome from basically spiraling out of control. Like, think about human chromosomes like a cup of water.
Okay, I like that analogy.
So if your mom gives you a full cup of water, which is 46 chromosomes, and your dad gives you a full cup of water, also 46 chromosomes, fertilization would logically result in, well, an overflowing cup.
Right, a massive spill.
Yeah, you'd have a zygote with 92 chromosomes.
And if you follow that logic to the next generation, you'd hit 184.
Which is just crazy.
Then 368. The cell nucleus would literally just pack with DNA until it became totally non-functional and burst.
A microscopic explosion.
Exactly. It's an exponential explosion that obviously does not happen.
So how does the body circumvent that math?
Through a really strict structural shift between what we call diploid and haploid cells.
Okay, diploid and haploid.
Yeah, so every normal body cell or somatic cell in you right now is diploid. Di meaning two.
Like a pair.
Right, you have two full sets of chromosomes, making 46 total. But your gametes, your reproductive cells, they have to be haploid.
Just the one set.
Just one set of 23. The challenge is that early cytologists back in the 1880s, they had no idea how this actually happened.
They were just looking through, like, really primitive microscopes, right?
Yeah, and noticing that reproductive cells seem to have only half the visible chromosome bodies compared to regular tissue cells.
And some of them actually thought it was just a bad dye job on the microscope slide, didn't they?
Oh, absolutely.
Like they were just miscounting because the stain was too faint?
They completely suspected it was visual artifact. But then August Weismann steps in around 1885.
The theorist.
Yes, he makes this brilliant theoretical deduction. Working on his germplasm theory of heredity, he argued from pure logical necessity.
Okay.
He reasoned that if an egg and sperm each carried the full chromosome complement, the number would double every generation, destroying the organism.
Right, the overflowing cup.
Exactly. Since the number remains stable, a reducing division must exist somewhere in the reproductive organs to keep the chromosome count constant.
That's so cool. He reverse engineered the logic of life purely on math.
He really did.
But someone actually had to find the visual proof. Which brings us to Eduard van Beneden and a highly convenient little parasite.
Right. Van Beneden was a Belgian embryologist, and he was studying the roundworm parasite Ascaris megalocephala.
A roundworm.
Yes, and this worm was an absolute gift to 19th century microscopists because its cells naturally only contain 2 or 4 chromosomes.
Which is perfect. You don't need a high-tech, modern lab to reliably count to 4.
Precisely. You really can't mess that up.
Yeah.
Van Beneden could clearly, visually confirm that the egg and sperm nuclei of this worm each contributed only half the number of chromosomes seen in his ordinary body cells,
Wow.
and that the original number was perfectly restored at fertilization.
Mm.
So Weismann's logic was confirmed.
That is so elegant.
It is. And eventually, in 1905, two British cytologists named Farmer and Moore, they just got tired of using these clunky descriptive phrases.
Right.
So they coined the term meiosis, originally spelled with an A, with the Greek word for diminution or lessening.
Okay, so we have the math problem solved. We know why the chromosomes have to be halved.
Right.
Now we need to look at the mechanics of how this actually happens, because it is not just a simple halving process.
Oh, not at all.
It is a massive genetic reshuffling. And I want to bring up a major misconception here, because when I first learned about cell division, I figured, well, regular cell division is mitosis.
Yeah.
So meiosis must just be mitosis happening twice, like a photocopy machine.
The old photocopy analogy.
Yeah. The cell hits copy, divides once, then just hits divide again without copying. I mean, that makes intuitive sense, doesn't it?
It sounds intuitive, yeah, but it is fundamentally incorrect.
Really?
Yes. Mitosis produces exact genetic clones,
Mm.
essentially identically replacing worn-out skin or liver cells.
Right.
Meiosis deliberately avoids cloning. The structural mechanics are profoundly different.
So walk us through the actual process then. What is the cell doing differently on a microscopic level?
Well, it happens in two distinct stages, meiosis the sect and meiosis the second. Before any of this starts, the cell replicates its DNA once. So you have 46 chromosomes, but each one is now made of two identical sister chromatids zipped together.
With the shape of an X.
Exactly. Think of an X. 92 chromatids total, but it's still counted as 46 chromosomes.
I'll add it.
Now, meiosis IO is the reduction division. This is the stage mitosis never does. The homologous chromosomes, meaning the maternal copy of, say, chromosome 7 and the paternal copy of chromosome 7, they actively seek each other out in the nucleus and pair up.
Wait, they literally zip together?
Yes, through a highly complex protein scaffold called the synaptonemal complex.
Wow.
You could think of it like a microscopic zipper that binds the maternal and paternal chromosomes perfectly side by side.
That is wild.
Then the cell pulls those intact X shapes apart into two new cells. The maternal chromosome goes one way, the paternal goes the other.
So they don't split the X.
Exactly. The sister chromatids stay bound together. Each new cell now has exactly 23 chromosomes. The genome has officially been halved.
Okay, and following that, meiosis the second begins.
Right, and this is the equational division, which physically resembles mitosis a lot more.
Okay.
Those two new cells divide again, and this time the X actually splits. The sister chromatids are finally pulled apart.
Leaving you with what?
You end up with 4 haploid cells. But the crucial detail that defines the whole process is that there is absolutely zero DNA replication between meiosis the first and meiosis the second.
Okay, that perfectly explains the halving part. But earlier we talked about reshuffling.
Yes, the genetic casino.
Exactly. And so far you've really just described a sorting mechanism.
Right, but the reshuffling is what makes you unique from your siblings. Remember when those homologous chromosomes are physically zipped together in that protein scaffold during meiosis the second?
Zipper thing?
Yeah.
Right.
While they are zipped up, they do something incredible. They exchange physical segments of DNA.
They swap parts.
Yes. This is called crossing over, and it happens at physical connection points called chiasmata. The zipper actually breaks the DNA strands and resplices them onto the opposite chromosome.
So they are literally trading chunks of genetic code.
They are. A human gamete usually has one to a few of these crossover events per chromosome pair.
Wow.
By the time they separate, the chromosomes aren't exclusively your mom's or your dad's anymore. They are a brand new, novel mosaic of both.
Okay, here's where it gets really interesting. Because for a long time, scientists knew about this shuffling mathematically thanks to early work with fruit flies.
Right, Thomas Hunt Morgan's lab.
Yeah, but they hadn't actually seen it. The genetic math was abstract until 1931 at Cornell University with Harriet Creighton and Barbara McClintock.
You know, the landmark maze experiment. That is a classic.
It is. They were working with corn plants, specifically maize that had visibly distinct, physically marked chromosomes.
Yeah.
And they proved that when genetic traits recombined in the corn, the physical, microscopically visible chromosome structures had literally swapped segments.
This is amazing.
They could visually verify the physical exchange of material under a microscope.
Which, an astounding technical achievement for 1931.
Yeah.
I mean, McClintock eventually won a Nobel Prize, albeit for her later work on transposable elements.
Right.
But this experiment firmly proved crossing over wasn't just abstract genetic theory. It was a physical tearing and recombining of the biological blueprint.
But wait, crossing over is only one layer of the reshuffling, right?
Yeah.
What about independent assortment?
Oh, right. The second layer is sheer mathematical probability.
Okay.
When those 23 pairs of chromosomes line up to be separated in meiosis SF, they do so totally randomly.
So they aren't organized.
Not at all. The maternal and paternal copies don't all line up neatly on one side or the other like two opposing sports teams.
That would be too simple.
Way too simple. It's an independent coin flip for every single pair. The math on that alone is 2 to the 23rd power.
Which is a massive number.
It yields over 8 million distinct possible chromosome combinations in a single human gamete. And that is strictly from how they line up.
Over 8 million combinations. And that's before you even factor in the physical crossing over we just discussed.
Exactly. When you combine independent assortment with the infinite variability of crossing over, the number of genetically distinct gametes a single person can produce is, for all practical purposes, limitless.
It's mind-blowing.
Every individual sperm or egg is a complete original.
Which answers the sibling thought experiment perfectly.
It does.
However, this raises a major anatomical assumption. If you're listening to this, you might logically assume that the way the human body makes sperm and the way it makes eggs are just mirror image versions of the same cellular process.
That assumption definitely makes sense on paper.
Right.
But biologically, the asymmetry between the two is staggering.
I want to explore that asymmetry. Let's look at spermatogenesis first.
Sure. So spermatogenesis is continuous and symmetric. From puberty onward, a male's body is constantly running this meiotic cycle.
Just a constant assembly line.
Exactly. It takes about 2 to 3 months from start to finish, and every single meiotic event cleanly produces 4 perfectly equal, functional, highly motile sperm.
A constant symmetrical biological factory.
Yes.
But oogenesis, the creation of eggs, doesn't follow that pattern at all.
No, oogenesis is discontinuous, and it is fiercely asymmetric.
How so?
When the cell divides, it does not make 4 equal eggs. It hoards almost all the cytoplasm, the organelles, and the nutrients into one massive functional egg.
Wow, okay. And the other cells?
The rest of the genetic material is essentially walled off and discarded as these tiny, non-functional packets called polar bodies.
Wait, why throw away 3 perfectly good cells? Why not maximize the reproductive output like with sperm?
Because a newly fertilized zygote has to survive for several days, dividing and traveling down the fallopian tube before it can implant in the uterus and draw nutrients from the mother.
Ah, I see.
It needs a massive internal energy store just to survive that journey. By sacrificing 3 potential cells, the body guarantees that the one remaining egg is large enough and nutrient-dense enough to sustain early life.
That makes total sense from an evolutionary survival standpoint.
It's incredibly efficient in its own way.
But the timeline of eugenesis is the part I find most difficult to wrap my head around.
Oh, what's fascinating here is the concept of dictate arrest.
Dictate arrest. Okay.
In a female fetus, the cells that will eventually become eggs, the oogonia, actually begin meiosis before the baby is even born.
Wow.
But they don't finish the division. They hit a biological pause button right in the middle of prophase I.
Wait, where is that again?
That's the exact stage where the chromosomes are paired up in that zipper structure we talked about, and they stay arrested there.
For how long?
Decades.
Decades.
They only resume and complete the division just before ovulation. This means that an egg ovulated when a woman is 40 years old has literally been frozen mid-division since before she herself was even born.
A 40-year pause in the middle of a delicate cellular division. It's astonishing that the structure holds up at all.
It is a biological marvel.
And the body discards most of them during this wait, correct?
The die-off is immense, yeah. Around mid-gestation, a female fetus has roughly 6 to 7 million oocytes. By the time she is born, that drops to 1 to 2 million. By puberty, it's down to a few 100,000.
That is a huge drop.
And over a total reproductive lifespan, only a few hundred will ever actually unpause and be ovulated.
So what does this all mean for you listening at home? We have this incredible machinery, but biology isn't perfect.
Right.
How does a microscopic, decades-long pause actually affect a person's life or prenatal care or clinical fertility?
Well, it leads us directly to the clinical realities of human reproduction, specifically a mechanical failure called non-disjunction.
Okay, non-disjunction.
Remember how those homologous chromosomes are zipped together in meiosis III?
Yep.
Non-disjunction is what happens when they fail to separate cleanly. Both copies of a chromosome get pulled into one new cell, leaving the other cell with none.
And is this mechanical failure tied to that dictyate arrest, the 40-year pause?
It is intimately tied to it. The cohesion proteins holding those chromosome pairs together naturally degrade over time,
Like old rubber bands.
exactly like old rubber bands. The longer an oocyte sits paused in prophase I, the more those structural proteins weaken.
So when they finally wake up.
Right, when the cell finally unpauses decades later and the spindle fibers try to pull the chromosomes apart, the physical risk that they stick together or separate incorrectly is much higher.
Which is the primary mechanism behind aneuploidy, the condition of having an abnormal number of chromosomes.
Exactly. The most well-known clinical outcome of this is trisomy 21, or Down syndrome, where a zygote inherits an extra copy of chromosome 21.
Right.
This specific condition was first linked to an extra chromosome by the French geneticist Jérôme Lejeune in 1959. And because of that protein degradation over time, the incidence rises sharply with maternal age.
Can you give us a sense of those numbers?
Sure. It goes from about 1 in 1,000 live births from mothers around age 20 to roughly 1 in 100 or higher for mothers around age 40.
Now, I need to pause here because our sources highlight a really vital historical point about how this data was used.
Yes, this is so important to clarify.
There has been a massive public health narrative built around this over the decades, presenting meiotic errors as solely an older mother problem. And we should just state for the record, we are solely reporting the biological data outlined in the sources here. We're not taking any social or political stance.
Absolutely.
But the historical texts make it incredibly evident that oversimplifying this into a maternal-only problem resulted in decades of skewed, really stigmatizing public health messaging.
It did. We must be extremely clear about what the biological and historical context actually reveals here. That widespread messaging masks a very nuanced biological reality.
Right.
While the maternal age effect is a proven mechanical reality, due to that oocyte arrest, meiotic errors are absolutely not exclusively a maternal issue.
They can happen anywhere in the process.
Exactly. The data shows a very real contribution from paternal-side meiotic errors during spermatogenesis, as well as post-fertilization mitotic errors where the early embryo divides incorrectly on its own.
So the biology is undeniably more complex than the historical stigma suggests.
Way more complex. And understanding those precise mechanical errors is exactly why modern clinical tools exist.
Like IVF screening.
Exactly. When IVF clinics use PGTA pre-implantation genetic testing for aneuploidy, they are literally extracting a few cells from a blastocyst and counting the chromosomes to screen for these exact meiotic segregation errors before implantation.
It's the same fundamental science behind NIPT, right? The non-invasive prenatal testing that so many expectant parents use today.
It is. NIPT analyzes cell-free fetal DNA floating in the maternal bloodstream just to check the math of meiosis.
It is just incredible what we can do now.
And furthermore, this understanding of meiosis is now heavily bleeding into modern oncology, which is wild.
Wait, oncology, like cancer research?
Yes, researchers have discovered that some malignant tumors actually abnormally reactivate genes that are usually strictly restricted to meiotic germ cells.
But why would a cancer cell want to reactivate reproductive genes?
Because meiotic genes are designed to do things adult somatic cells shouldn't do.
Like what?
Like evade natural cellular checkpoints, achieve cellular immortality, or rapidly shuffle DNA to adapt.
Oh, wow. So the tumor is basically stealing the meiosis toolkit.
Exactly. When a tumor activates them, it's hijacking that specialized machinery to survive and grow.
That is terrifying, but also genius.
They are called cancer testis antigens, and immunologists are currently exploring them as prime targets for highly specific cancer immunotherapies. Because healthy adult tissues shouldn't be expressing those proteins at all, making them a perfect target for the immune system.
That fundamentally changes how we view the utility of this entire process.
It really does.
But it brings up a larger issue. If this meiotic process is so metabolically exhausting and it requires such an insane amount of energy, and frankly, it's so inherently prone to tragic errors, as we just discussed,
Right.
why did evolution select for it in the first place? Why haven't we evolved a safer, easier way to reproduce?
This raises an important question, and it is actually the center of massive, completely unresolved debates in evolutionary biology right now.
Really?
Yeah. Why did meiosis and, by extension, sexual reproduction evolve in the first place?
I mean, I would assume it's just for the genetic diversity we talked about earlier, to mix things up.
Well, that is the classical view. It's been championed in various forms since August Weismann.
Okay.
The idea is that meiotic recombination generates the immense genetic diversity needed to speed up adaptation. Shuffling the genetic deck every generation helps the species survive changing environments, outpace evolving parasites, and purge harmful mutations.
That seems perfectly logically sound to me. What could the rival view possibly be?
The rival view is known as the Bernstein DNA repair hypothesis.
Bernstein DNA repair.
This theory argues that meiosis did not evolve for diversity at all.
Not at all.
No, it argues meiosis evolved primarily as a mechanism for early single-celled organisms to repair severe DNA damage.
Let me make sure I'm following this. How does shuffling DNA act as a repair mechanism?
Think about a double-strand break, where a chromosome is literally snapped in half by radiation or free radicals.
Ouch. Okay.
That is a lethal error for a cell. You can't just glue it back together because you've lost the blueprint at the break site.
Right, the information is gone.
Exactly. The Bernstein hypothesis suggests that crossing over, finding a homologous chromosome and zipping them together originally evolved so that the damaged cell could use the intact partner chromosome as a physical template to accurately copy and repair the break.
Oh, I see. So the repair is the main event, and all that wonderful genetic shuffling and diversity is just a secondary, accidental byproduct.
Exactly. A happy accident of a massive cellular repair job.
That is wild.
The debate remains totally unresolved today because both the diversity benefits and the repair benefits are real. Biologists are simply arguing over which one was the original evolutionary spark.
Well, if the underlying purpose of this machinery is still up for debate, it makes sense that the execution is completely different across species.
Very true.
Which brings us to the concept of heterochasmy in our sources.
Ah, yes. Heterochasmy is a completely baffling observation.
The great word.
It is the scientific term for the fact that recombination rates, the actual frequency of crossing over, differ wildly between the sexes.
How so?
In humans, females generally have a much higher overall recombination rate during equigenesis than males do during spermatogenesis.
But males tend to recombine more specifically at the physical ends of the chromosomes, right?
Yes, the localized patterns are completely inverted. And no one genuinely knows why this sex difference exists, or why it varies so unpredictably across different animal species.
It's just a blank spot in our knowledge.
Completely. If it were strictly just a DNA repair mechanism, or strictly for diversity, you'd expect the rates to be somewhat uniform. It remains a completely open biological mystery.
I love that we don't have all the answers yet. It keeps things exciting.
Definitely.
But there is one more mystery, or maybe a rebellion, that we have to discuss. The forces that actively cheat this entire system.
Oh, if we connect this to the bigger picture of evolution, meiotic drive is terrifyingly brilliant.
Meiotic drive.
Yeah. Everything we've discussed so far relies on meiosis being a fair mathematical lottery, a 50-50 shot for any gene to make it into a gamete.
Right.
But meiotic drive is when a specific gene variant figures out how to rig the game.
How exactly does a gene rig cell division? That sounds impossible.
The classic, most well-studied example is in mice. There's a gene variant on chromosome 17 called the teeth haplotype.
The teeth haplotype.
Yes, it literally manipulates sperm function in males. During spermatogenesis, this gene variant produces a poison that affects all the sperm, but it also produces an antidote that it only gives to the sperm carrying the teeth haplotype variant.
Wait, it poisons the other sperm. It destroys the competition from the inside.
It actively disables the non-carrying sperm, ensuring that males carrying this variant transmit it to over 90% of their offspring, rather than the fair 50%.
That is incredibly sinister.
It completely shatters Mendelian inheritance. And the wildest part is, it does this even though having 2 copies of the teeth haplotype is deeply harmful to the mouse's overall survival.
So it doesn't even care if it kills the host.
Nope. It breaks the fair lottery rule purely to ensure its own transmission, regardless of the health of the organism.
Well, we have covered a massive amount of ground today on the anatomy of how we are made. Let's do a quick recap so you can walk away with the absolute essentials.
Sounds good.
First, meiosis is definitely not just mitosis happening twice. It is a highly specialized 2-step division. Meiosis-ios halves the chromosome count by separating paired homologous chromosomes, and meiosis II separates the sister chromatids with 0 DNA replication happening in between.
Second, through the physical crossing over of DNA and the mathematical independent assortment of chromosomes, meiosis ensures that every single gamete is a mathematically unique mosaic of genetic material.
Third, the biological timelines for making these reproductive cells are radically different. Sperm are produced continuously from puberty, while human eggs are paused in prophase I for decades. And it's that very pause, the dictate arrest, that causes structural cohesion proteins to degrade, explaining the mechanical reality behind chromosomal conditions like Down syndrome.
And finally, while this intricate cellular machinery is the engine of our genetic diversity and survival, it is not an invincible system.
Not at all.
It is vulnerable to mechanical decay, evolutionary debate, and active genetic sabotage, as we see with the mouse teeth haplotype.
Which leaves us with a rather unsettling final thought to ponder today.
Oh, absolutely.
We know for a fact that mice have gene variants that actively cheat the fundamental fairness of meiosis, destroying competing cells to ensure their own survival at the expense of the larger organism.
Yeah.
It forces you to wonder, what if there are undiscovered selfish genes hiding in the human genome doing the exact same thing right now?
Scary thought.
What if fragments of our own DNA are quietly rigging our evolutionary lottery, spreading through the human population by breaking the very rules of genetics we just spent this entire session discussing? It is a wild, chilling thing to consider.
It really makes you rethink how much control we actually have over our own biology.
Thank you so much for joining us on this deep dive into the microscopic origins of who we are. As always, keep asking questions and keep learning.